Hey everybody, and welcome to the 2020 Science for Shishi Symposium. Keeping in theme with 2020, this year's symposium is going to be completely online, so you can enjoy it from the comfort of your own home. So get ready. My name is Ansh, and you're watching Disney Shoot, wrong script. Rohan, roll the intro.
The pace of progress in biology creates a foundation that naturally gets picked up by the biotech and pharmaceutical industry to solve rich world disease. This is attractive science. It's science that people want to work on.
This form of science is something people were naturally drawn to before the actual term was created. It was not until 1919 when the first occurrence of the word biotechnology was heard, which was brought upon by the Hungarian engineer Karl Erki. So the first question we asked ourselves is what biotechnology actually is. So biotechnology is the branch of sciences that uses recombinant DNA techniques for practical purposes. So it's the form of technology that focuses on the utilization of biological systems and living organisms to produce and develop desired products. So you have both traditional and modern biotechnology. Traditional biotechnology would involve the production of bread. So say you have yeast as a living organism, it is used to create the desired product, which is bread. So it's utilizing this living organism as a natural source. Additionally, you have modern biotechnology. So modern biotechnology is, on the other hand, is the implementation of these advanced mechanisms to modify these biological organisms and biological systems.
Moving on from what biotechnology is, let's get into the history. From our past to now, the progress of biotechnology can be classified into three distinct eras: ancient, classical, and modern biotechnology. Ancient biotechnology prior to the 1800s dealt with the utilization of domestication, fermentation, farming, and even crossbreeding. Classical biotechnology, from about the 1800s to the mid-1950s, was when many great scientists such as Gregor Mendel, Friedrich Miescher, and Alexander Fleming made huge advancements and identified the transferred genetic material, identified the genetic material itself to be DNA, and even discovered antibiotics. Modern biotechnology is from the mid-1950s to now. Through the visualization of the double helix to the figuring out the entire genetic code, the field of biotechnology has become revolutionized. Huge advancements have even made live clones possible, such as carbon copy the cat, that are complete genetic copies of their parents. Back to you, Rohan.
So being part of the modern biotechnology era, this field is being heavily researched, resulting into a surplus of advantages. And this is especially true with approximately $20.4 billion being invested into the research and development of biotechnology in 2005, making it one of the most research-intensive industries in the world. In terms of advantages, it improves our everyday lives as it is implemented to improve crop yields, cut costs, and even reduce pesticide applications. It makes our food safer by creating products that prevent animals from being induced with the E. coli bacteria, thus humans not contracting it either. Biotechnology is also responsible for various medical diagnostics that protect the blood supply, for example, from the AIDS virus, and additionally prematurely detect various conditions to lead to successful treatments. In terms of drawbacks, the extensive and the excessive use of biotechnology, for example, in genetically modified plants, this can lead to the loss of biodiversity and the loss of genetic diversity, as well as soil depletion and crop yields. So moving forward, biotechnology is an expansive topic that can be further divided into smaller subtopics. There's gel electrophoresis, DNA probing, restriction fragment length polymorphisms, and even polymerase chain reaction. Out of all these discoveries and processes, the most enticing subject under biotechnology is gene cloning. Moving on, we have invited some special guests to help us gain some insight into what gene cloning is. Please welcome our neighboring 2020 presidential candidates, Donald Trump and Joe Biden.
Biden, crazy weather we're having, isn't it? One time it was raining, and I just made it stop. I'm so amazing, what can I say? Everybody's talking about it.
Sure, Trump, but we have more pressing matters to talk about. We were invited to talk about gene cloning.
Yeah, of course I know what gene cloning is. It's an actual science, unlike that climate change nonsense. But why don't you start talking about it first?
Okay, Trump, whatever you say. Gene cloning is a process in which DNA is manipulated for the production of many identical copies of a gene or a different segment of DNA in foreign cells. Specifically, the concept producing clones has many applications such as genotyping, mutagenesis, sequencing, or heterogeneous expression of
Yes, you can waste time talking about that, or you can talk about the current world, where gene cloning is predominantly used for isolating long or unstudied genes and protein expression. I'll create so many more jobs. I'll tell you, American jobs.
You're spot on, Trump. Something you're not very good at. Just like your dance moves.
I'm amazing at dancing. I'm amazing at everything, Biden. Wish I could say the same for you. The future, the potential for gene cloning is immense. The process can be utilized to produce a protein product, like insulin, in large quantities. This creates an inexpensive source of pharmaceutical drugs, a field in which I have many friends. And bacteria can be engineered to cleanse environments as well as engineering them to consume toxic waste. In terms of research, producing more than one copy of a gene can be a very beneficial tool.
To add on, by way of gene therapy, non-functioning genes can be replaced with a functioning gene and a gene in a... my bad, sorry about that, folks. To add on, by way of gene therapy, non-functioning genes can be replaced with a functioning gene in a human cell. Due to the multiple errors associated with this potential for gene cloning, like humans becoming sick and non-functioning protein after a short period of time, this technique still needs to be heavily researched for it to be mastered.
Biden, the research related to gene cloning is mainly provided through the studies and experiments conducted on genes, specifically E. coli. This bacteria has many reactants that have been optimized for its cloning process. Gradually, all the scientists are moving their experiments on live subjects, like insect cells. I've seen this. I've sort of witnessed it. In fact, 2 cases I've witnessed this.
Wow, that's great to hear. I'm so excited for the future of gene cloning and what it has to provide for our great nation.
I could have said it better myself. I think it's time for my lunch break. Let's send it back to those 2.
Definitely. Thanks for everyone tuning in to this interview and discussion between Trump and myself, the future leader of this neighboring great nation. And have a good one, folks. Hashtag vote Biden for 2020.
Thank you for that great segment, Biden and Trump, but now let's get back to the explanatory bit. Gene cloning is a highly intricate and specific process that involves the replication of a single gene or a section of DNA selectively. I would love to draw it out for you all, but my hand's a bit sore. Mind lending me a hand, Ansh?
Of course, so now we're going to talk about the gene cloning process. It is very intricate and highly specific and is used to replicate a single gene or section of DNA that is chosen very selectively. The first step is to use restriction enzymes to isolate and cut a selected portion of DNA. Let's take the gene that codes for insulin production as an example. In the next step, this cut portion of DNA is then transferred into a plasmid, which is a circular extrachromosomal piece of DNA, usually found in bacteria. This is done via the help of DNA ligase enzymes that join the ends. At this stage, an antibiotic resistance gene is also inserted into the plasmids to help their identification later on. Next, the plasmids are inserted into a vector, which is another cell that will carry the plasmids to help replicate the gene. The vector in this case is E. coli bacteria. The plasmids are encouraged to enter bacterial cells via a shock peak. Now, the altered bacteria are cultured in dishes that have appropriate growing nutrients as well as antibiotics. This allows only the cells that have taken in the plasmids to survive, as they have a copy of the aforementioned antibiotic resistance gene. As the bacteria reproduce asexually via a process called fission, they also make copies of the plasmids and subsequently the selected DNA in them. As the bacteria grow, they express the traits not only on their genome, but also the traits on plasmid DNA. This can be used to achieve many goals, for example, the production of a specific enzyme or molecule, such as the insulin in this case.
So gene cloning is a complicated process that requires distinct knowledge and access to resources. For example, a molecular biologist. This involves studying the inner workings of a cell and their biochemical processes. Additionally, gene cloning requires intricate techniques to form recombinant DNA in one molecule. So having the enhanced knowledge of cell biology, and the associated chemical reactions is required to successfully achieve this, which is what a molecular biologist is capable of. There is also genetic engineering. To achieve recombinant DNA is a difficult task, which a genetic engineer excels in. The manipulation of genes and the alteration or cloning of genomes is the specialty of a genetic engineer and the most important tool in gene cloning. Last but not least, embryology. Gene cloning is capable of forming an embryo that is identical to the genome of the parent. The embryology, or the formation of an embryo, is vital for understanding the intricacies of gene cloning and the effect it has on the organism.
With all this knowledge of gene cloning, some actual progress is being made, as evident by the various applications of gene cloning. For example, the same researchers who produced Dolly the sheep have also produced other cloned sheep that have the human gene for a protein that helps with blood clotting. Additional work is being done in order to isolate this protein from the milk to help patients that have genetic disorders. Similarly, gene cloning can help aid a process called heterologous protein expression, in which large amounts of a specific protein are made via the help of a self-replicating genetic element, similar to how insulin was produced by the E. coli bacteria in a previous video segment. Secondly, natural variations in gene expression allow for increased yields by certain members of the population. Animals that produce milk or meat in relatively higher quantities, for example, can be chosen and cloned in order to increase the production of the entire farm. Another application of human gene cloning is the creation of embryonic stem cells that have nearly limitless potential in the treatment of all kinds of diseases. The initial zygote that is created by the human gene cloning process has the exact same DNA as the parent. And since embryonic stem cells can differentiate into any other type of cell in the human body, the applications in medicine are profound, once we make it past some of the ethical concerns, of course. With all this newly attained knowledge and understanding of gene cloning, it may seem that it's too good to be true. This is why we've created an online forum for you guys to share your thoughts on some of the moral implications of the gene cloning process.
Our comments are already posted, but we want to hear what you all have to say. It's as Gates mentioned, this is attractive science. It's science people want to work on.